Why are we here? Why does SRF work so hard to make this conference happen? Because we're all parents of sick kids, and we want our kids to get better because the existing solutions, frankly, aren't acceptable. And that is why we do what we do. That is why we pound the table. That is why we bug you and ask you to sign up for trials and go far away to do natural history study collection, all the things. And really, it comes down to this next session where we have pulled no punches because we want to understand what's it going to take to get therapies into our kids. You got a taste of it the last time I got motivated to stand up here when people said, what are you going to measure? Like, how do we actually do this? And for this, we called in one of the big guns, Dr. Anna Mingorance. And I met Anna at AES years ago through my friend Don Blessing, who at the time was at Zogenix. And Don was like, you got to meet Anna. And I think I was like 2 years into this and I was clueless. I was like, where am I? What do all these people do for a living? And Anna has been kind and wise with me ever since then. And at no matter what time or whatever she was on the planet, when I would send her an email, she would reply. And she is a consultant to industry. She's an advisor. We have never paid her a dime, and yet she continues to be very interested in Syngap, always take our calls, and always agree to do things like run this next session, which we all need to listen to and we all need to focus on. So what's next to get therapies into our kids? I think as you've heard in this session, as compared to a year ago, we're talking about a lot of molecules. Zach is already dosed, Dr. Grinspan, pardon me, is already dosing kids with Ravicti, right? We're talking about NAL, we're talking about nortriptyline. Dr. Novak has a list of a bunch of data that I haven't even seen yet. I mean, there is so much going on. And then we have all these precision therapies while we have this paucity of patients of countable motor seizures. So things are getting very real. And I think we need to focus on what's next so we can focus our time and attention. Because unlike the early days, when I was like, hey, Syngap, I now have this many grant proposals on my desk and this much money, right? Whereas before I had this much money and this many grant proposals. So we got to focus, and we got to focus on the clinic. And I'm so glad to see Dr. Andrade here, who is working on the adults, because we get more and more adults diagnosed every day. And we want therapies that'll help all our kids who are 30 and 40 and 60, right? So with no further ado, this is a really important session. Thank you for putting up with us today. There are drinks later tonight, so hang in there. And thank you again for being here. Over to you, Anna.
Well, I'm gonna go back there.
Okay.
So I had a few slides to start, and then I'm going to call a panel of biotech speakers because they will help us see from the other side how they see the field and where are the gaps and what's next. I guess it's the green button. There are too many buttons, and I'm still traumatized from the fly presentation. I know it works, and I can see it there. So I actually changed a bit the name of this session more about readiness. We're always talking about research readiness and trial readiness. So where should we be? Where are we now? And essentially, talking about readiness is like a huge checklist of things that we should do to make development for therapies for Syngap super easy. How many have we checked already? So I took the liberty to score this myself. This is my day job. So I also love Excel sheets, Catherine. So this is too much text, and how we're going to do. Anyway, we're not going to read it. We're just going to know that for the research readiness, later I'll show you trial readiness, we need to answer a lot of things, and this is even just a very simplified thing. So is the disease on the radar of everyone? Based on that is, do we have enough patients? Is it genetic? We need to know what to target, all of that. Feasibility to study it is the preclinical tools. We've seen a lot today about preclinical tools. Do we have cells? Do we have mice? Do they have phenotype? We have antibodies for all that. Tractability about the preclinical part is, do we know that the disease can be targeted in older animals? Do we know what's the minimum expression we need to increase and where should we express it and all of the isoform thing? And then you also need to have strong patient foundations that will help you build all that. So if I had to score all that, I think we're doing quite good on this side. There are a little bit of gaps on this side. Still, we're talking about those isoforms, about the minimum expression, but that's normal for most diseases to know. This is the start and the big strengths. You heard it so many times today, and I hope the biotech panelists will repeat it today again. Like, this is one of the huge strengths of this disease, the foundation and the patient community it has behind. So if I look at that, how are we doing in research readiness in SYNGAP? I think we're very good as all the diseases that already have disease-modifying therapies in trials. It's not because of not being ready. I don't know why. We'll ask them. Clinical trial readiness, same, huge list of things we should be doing. Do we know the clinical picture of this disease, how it looks? For regulatory folks, this is all the disease concept model. Did you talk to FDA? Do they understand the concerns, natural history, all of that? Regulatory, here I think I scored them one by one, yeah. And we're good here. Regulatory precedence, is the disease acceptable because you have a CD10? Do we know it's acceptable because of orphan drug designations? Have we had trials phase 2? Have we had phase 3? We still haven't had the phase 3, working on all that. Endpoint, so what are we gonna look and how much is good enough? Again, you need to, often the evidence comes from prior trials, so it's kind of a catch-22. If you haven't run the trials, how do you know you have them? And yeah, this is another one that I give it, like working on it in particular due to biomarkers, but very promising EEG biomarkers coming from CHOP, by the way. And then recruitment. Do we think that if we had a therapy for a trial, a large phase 3 trial, can we do it? This, again, because this is the strength of your community. If there is a disease where I have no question that if someone were to run a big phase 3, we're going to pull it off, that's SYNGAP, quite clear. So if I look at all of the trial readiness, we're still missing a few things, but so do all of the other diseases that are already having clinical trials with gene therapies, with ASO, so it's not different. And what I mean with all of this, my big message is, we're going to be talking about gaps right now with the biotech folks. And we're going to be talking about those things that are missing. And I don't want you to think that those things are missing to be good enough. No, this is more than good enough. This is way above where all the diseases were, where they had those treatments in trials. We're talking about how to make it even faster, because we want to make all that even faster. But yeah, that's the message. And again, what we wanted to do with this final session, which is, yeah, the last one, was to recap. We've had the full day, all the academics present on the progress, and I wanted to hear now from people from industry. All of them have put drugs into trials. Some of them have put drugs in the market. How they see that readiness, highlight some of the things that are working, but in particular, advice on those are the things we can do to make it faster. So the gaps and the areas for improvement. But that's what I wanted to frame it as, but that's just to make it faster. Don't think that that's because we're falling short, we're not. And it was important to have that version from biotech. And I think I'm going to start with, with Chris, since you're here. If you could tell us, so I guess introduce yourself a bit at Praxis and how you see the field today and some of those areas that you think are gaps or advice. We're going to be taking notes.
All right. All right. So first, thanks for the, opportunity. So thanks for the opportunity. I would challenge Mike when he says that we're doing, like, you guys have made this progress because you care about your kids and you don't think the current therapies are acceptable. Because if that were simply the case, all community groups, all family foundations would move at this pace. And it's just not the case. I think it's complete BS. I think it's the culture that you guys have. And it's really, it's amazing. So for those of us trying to put things in the clinic, it's really great to have that as a partnership. Preclinically, Sana asked me to speak a little bit preclinically. I think, this gene, this field, there are challenges with splice isoforms, there's challenges with the type of modality that we're going after. So Praxis has an ASO program that we've talked about, and it's delivery. That's going to be a challenge because right now ASOs are intrathecally delivered. There's a comment on challenging the field to get better ASO delivery. If you went to the, or those of you who were able to or could tune into the OTS meeting in October, that's all the space is about, trying to get ASOs in the brain and get them distributed. So there's a lot of smart people working on that problem, and it's a very difficult problem to solve. But having the research models available, having them available to industry, I can't stress how important that is to being able to accelerate progress.
So I think that's a great jumping off point to go to Greg, because earlier today we heard about how SRF helped create a mouse with a nonsense mutation so that a particular company could test their technology. And I would like to hear from you the other side of the story from the company. Why was your technology so unique that needed a specific animal, and how was it working with SRF?
No, that's a good question. I mean, I'll tell you a little bit more about what TAVR does. As we were told and know about, nonsense mutations account for about 10 to 15% of all genetic diseases. That's a lot of people. And when nonsense mutations are put in, as Ben so elegantly laid out, you end up with premature termination codons that increase nonsense-mediated decay of the message and decrease the translational read-through, and you get truncated proteins that cause disease. At TAVR, it's similar to the ACE tRNAs. We use what's called suppressor tRNAs to read through those premature termination codons and create full-length message. So we're modifying not only the anticodon, but we have second site mutations that we use where through screening and through artificial, through AI and machine learning to find the optimal read-through. And we're really interested in SYNGAP because when you look at what accounts for the disease nonsense in mediated nonsense mutations in SYNGAP, 35% are accounted for by mutations, nonsense mutations. Of those, only 2 premature termination codons or 2 tRNAs are necessary to account for 80% of the read-through. So we're working on one of those. And as Anna said, one of those is the mutation, the Q503X from the SRF Foundation along with Jackson Labs. And it's crucial for us to have these type of research reagents because for us to move forward, having these tools allow us to find out, do our tRNAs work? Do they have all the necessary function to alleviate the symptoms in a mouse and move forward with groups like the SRF and all of you who fund it, as well as JAX to help with the phenotyping and all of this. It's a big effort. And having this group and well coordinated and be able to provide these research reagents is invaluable. So thank you.
Thank you. And Justin, if you want to add to the research state readiness, what do you see as the strengths? What do you see as some learnings?
Yeah, I'm Justin Ichida from head of neuroscience research at BioMarin. We're a pharma company up in Mike's backyard, actually, in Marin County. And, I think aside from having this amazing foundation, I think the other strengths, of course, are the huge unmet need. I don't think we need to reiterate that, but there are also some of the facets of the disease that could enable us to know if a drug is working fairly early on in the disease, in the clinical trial, so that we don't have to ask more patients to enroll if we don't think it's going well. For example, I think the seizures, I know that only a certain subset of the patients will have those measurable seizures, but I think there's reason to believe that if you can modify those with a certain level of SYNGAP, you may be able to modify some of the other features of the disease, like intellectual disability. So I think that's something that is promising. Now, there are a lot of challenges. I think almost all of them were mentioned today. How much SYNGAP, for the ones that are trying to restore SYNGAP, how much SYNGAP protein is necessary? How reversible are the different symptoms of the disease? I think the animal models are telling us that there's some reversibility probably in the behavior and the seizures, and of course the repurposing studies as well may be suggesting that. But we don't know how many of them are reversible, and more importantly, we don't know what age of patients would be most responsive. And so when you think about how do you enroll the trial, do you want to enroll pediatric patients or older patients because they might be more tolerant to, any kind of safety issues that come up? I think that's a key question. And then I think beyond that, the characterizing the mutations. I think these, we talked about the missense mutations a lot today. Can we do studies where we, actually make these mutations, look at them in cells, and know if they're increasing SYNGAP function or decreasing SYNGAP function, to know that these are mutations that are causing the disease, but also 2, that we don't elevate ones that have like a toxic gain of function that we'd want to avoid. So those are some of the key challenges I foresee.
Thanks. So I'm going to go to the other half here and move a bit more clinical. And you highlighted that maybe seizures can be good as an early response signal, but we heard a lot today here that one of the negative sides of this disease compared to others is that there's not enough epilepsy, at least seizures of the countable type, to really use it for trials. And I'm going to go to Emma from Encoded because you've been working on Dravet syndrome, taking a gene therapy to trial, and that's the disease where we actually did have seizures as a good endpoint. But because you came into the clinic with a gene therapy, and a gene therapy needs to do a lot more than just reducing seizures, you almost have to move the disease from this is an epilepsy to this is a neurodevelopmental disease, and convince regulators to that, and even design trials. One of your phase 1/2 trials, I think the one in the UK, even has a non-seizure endpoint as the main epilepsy readout. No, main efficacy readout. So if you could tell us a bit that process of like Syngap became famous as an epilepsy, we're probably going to have to move it as a popular neurodevelopmental, because every time someone tells, oh, you don't have enough seizures, I said, that didn't stop Angelman, that didn't stop Rett. So how is the transition to go to the Angelman and Rett box?
Well, it's a tough question, and I think it's still very much a work in progress in terms of moving perception of these disorders from being seizure disorders to being, far more affecting the whole body. I think, in terms of what we've really learned in the Dravet space, I don't want to applaud you all because I think Syngap is doing, everything that the Dravet world has done to be set up for gene therapies really well. But I think really, understanding the natural history of the disease in the contemporary natural history in age-appropriate populations. So really studying the progression in young children, in adults, in all the different groups, so you can have that conversation with the regulators about what kind of change you anticipate seeing, because the challenge with the neurodevelopmental disorders is that often the regulators are still perceiving these as seizure disorders and not really understanding that there's a lot more needed. I think also one of the challenges we've had to face is the fact that in these disorders, the outcome measures haven't really been well defined. And so again, really understanding which outcome measures are most suitable for different age populations, and do they capture the kind of changes that you need to see within your children. I will say, I think there is definitely a move from regulatory authorities and ethics committees, particularly, that they really want to hear from the patient community. I think many people here will be familiar with the Patient-Focused Drug Development Initiative in the US, but certainly within the Dravet world, we've undertaken qualitative and quantitative work to understand the risk and benefit assessment, and really what the patient community is willing to undertake to advance these kind of complex medications. And the regulators and the ethics committees really do want to hear now. I think multiple different engagements we've had where committees maybe have had some concerns around invasive routes of administration with therapies that you can't just take away quickly. And the feedback from them has always been, well, what do the patients say? What do they consider to be reasonable? And I think that's a real change. But I think it is very, very positive for the community. It does mean that the companies who are doing this kind of work are going to keep coming to you and asking you about this kind of stuff. And of course, that does pose more of a burden, I think, on the patient community. I think more pre-competitive collaborations on that kind of stuff could really help, because a lot of it, it is just something that can benefit the whole community as a whole. So I think advancing things in that way that it doesn't have to conflict with companies' IP would be super helpful. And I do think that that's been something very, very positive within the CNS and the neurodevelopmental space that I haven't necessarily seen so much previously of people really starting to come together and work across disorders and across, you know, across companies as well to try and do that. I could keep going, but I'm going to shut up for now.
Thank you. No, but it's good if you can give it to Parisa. Yes, because Parisa from Stoke is one of those people that have a job that is to be that connection between the patients and the company, to bring the voice and the needs from the patients to the company and the questions from the company to the patients. So I think it's good if you can continue on that to see how that patient community can collaborate with industry through people like you.
Well, thank you. So Parisa Sanandaji, I head up global patient advocacy and policy at Stoke, and very happy to be here. But I really do want to echo the sentiment so far you've heard from the industry colleagues that, and I've said this to you all before, I've had the privilege of, this is my third conference with the SRF, and I've spoken to your board, and you all are quite advanced. You're quite sophisticated. There's a lot that you're doing that it's hard for me to add on to. And so what I would add on to, Anna, is actually to take on what Emma was just saying. You know, beyond the PFDD, we're moving into another era of patient-focused drug development with FDA. There are additional levers by which you can engage with regulatory bodies, both in the US and Europe, really across the world. And we know that they exist now. And you had a slide up here before around clinical trial readiness, and I'll tell you, SRF checks off a lot of those, right? They're doing a lot of it. And you all have developed this resource that I know your colleague, Lindsay Wizrak, who's not here today, I don't think, I haven't seen her, has developed and has shared with, again, going back to what Emma said, this pre-competitive landscape of other companies coming together. We belong to the Global Genes Corporate Alliance. You all are members of that as well. And this resource includes all the incredible work you're doing already. It captures the rare community that SYNGAP is made up of, all the different sort of pieces to your kids, your families. You include the partnerships you're currently engaging on. In that resource, you also include your research tools, your mouse models. I mean, these are super sophisticated. And I will tell you, very few patient groups whose, I think, lead candidate is in still very much preclinical development is doing this, is just deserving of a lot of applause. And it's a true pleasure to be sitting in this kind of a chair, because I didn't think this would happen this quickly. And you also include the therapeutic pipelines across all the different companies, and you have us all sitting here talking about it. So you're doing so much of it. And I think continuing to hear some feedback around what you can do to change regulatory policy is going to be an area that I know many of you are focusing on. I'm looking at a couple of folks here who are on the Hill and are out there advocating. So continuing to do that. So stay tuned. And for my industry colleagues, if you have patient advocacy folks on your teams, there are resources for you all to take on to do work like SYNGAP does. And frankly, this is a framework that SYNGAP is doing for other patient groups to share. So stay tuned for a white paper that comes out with Lindsay's work in it from Global Genes. And I'll leave that there.
And I think, Sojash, you also wanted to talk about some interactions with regulators, with patient groups.
Sure. I mean, I think a lot of what I was going to say has been said already.
Yes,
actually,
I know.
but
I was realizing.
But no, it's been a really nice conference for me. I'm a pediatrician by training and head of R&D at a small startup company. I think a couple of comments I would add that I think may have been missed are, it is really important to interact with regulators. It's important to interact with regulators early, in particular on the company side. And, you know, you saw from Ben Prosser's presentation earlier, there are actually a number of newish modalities that haven't really been in clinical trials. And the FDA and other regulators are really keen to hear about those early. And to build on Emma's point, if there are risks associated with them, you know, you don't want the FDA to overreact to those. So some information and from parents and families saying, okay, we understand the risks, but we would like to move forward anyway, I think is a really important thing to push forward with the regulators. I think it's important to talk to regulators outside the US as well, because sometimes there's a regulatory agency that's willing to get a program into clinic sooner than the FDA are. I've had that experience several times, actually. And so it's important to not just speak to the FDA, but, you know, maybe the MHRA in the UK or PEI in Germany, for example. I think it's important for the patient organizations to also familiarize yourself as you are doing with the technologies that are coming through, because what I hope would happen for this group in particular, say, is, within 2 to 5 years, you're going to have to make choices on, well, my child has SYNGAP, I want them to enter a clinical study. Which one should I do? Should I do the AAV gene therapy? Should I do the CRISPR? Should I do the ASO? And in order to make that choice, you need to be as knowledgeable as possible. And, it's a challenge, it's a problem. It'd be a great problem to have. And I think that's one thing, I hope 3 or 4 years from now, we're actually talking about the clinical trials and the dosing of patients. I think the final point I'll make that haven't been touched on today either, but I think is something that you as patients and families can get involved in is, policy developments, health policy, and, I hesitate to use the word politics, but getting involved at that level as well. The more from a rare disease perspective than from a SYNGAP specific perspective. The Orphan Cures Act is going through Congress at the moment, and, there's a lot of good information and good ideas about rare disease, clinical, preclinical, and scientific research. And if that bill can go through Congress, get passed with support from some of yourselves, I think that'll be really helpful and impactful for the rare disease community.
Thank you. And we still have some minutes left, and we've made sure that everyone had a chance to speak at least once. But if there is some question that you guys have for biotech, they're captive here. Should. Ooh, perfect. We have a microphone to Catherine.
Hi, I was wondering if any of you can tell us how do we go to the FDA? Do we go actually with an industry partner? Do we go by ourselves? What are the best practices there? You want to do it?
I mean, we can start. I mean, I think I already mentioned that there's the patient-focused drug development initiative. So the FDA does patient listening sessions. They do them both on, you as a patient advocacy organization could request a listening session and actually talk with them specifically about what it is living with SYNGAP and what you hope to achieve with the new therapies. They also have listening sessions about, gene therapies and the emerging risks and benefits associated with that. So they post that kind of stuff on their websites, and you can just submit a request to join and provide a comment. I do think also there are opportunities as organizations to go with companies. Usually it's just, one person from an advocacy organization, but it can be incredibly meaningful for a sponsor to have a patient advocacy representative there to really make sure that the regulators understand from that patient perspective what they are willing to tolerate. I think it's very different hearing it directly from a patient versus reading it in a document that a sponsor has written. And then finally, I do think the ethics committees and the regulators, they are looking at published literature now. So, doing a study, doing a survey, put it out, whether it's a poster or whether it's a full manuscript, but that can also be submitted along with any applications. And they do read them, they do ask questions on them, and they do listen to them. So I think there are a variety of ways, sort of directly and indirectly, to engage.
I will just add in a plug because it's pretty timely. So the Rare Disease Innovation Hub was recently launched at FDA. There was a public meeting in October. And so that is actually going to be a really direct channel. Its main purpose is to create, we were supposed to have a center of excellence for rare disease, and instead we got this hub, but they are hiring a real point person. So between Peter Marks and Patrizia Cavazzoni, this is something certainly we can follow up on across companies, but the Every Life Foundation is a great place to go to as well to really get a great answer on regulatory policy working group issues. You can sign up. Again, I would recommend a couple of the advocates from SRF sit on that regulatory policy working group. They meet quarterly, and they are already thinking about ways to engage the RDIH, the hub, next year. And I do think that's going to be really interesting. And just want to add on to your orphan cures piece. This is around a lot of impact that will, well, a lot of potential implications and unintended consequences of the IRA on early drug development and especially in rare diseases. And so certainly, again, going into these other groups, galvanizing with your other, DEE groups, the Dravet Syndrome Foundation is another one, but there is quite a bit around orphan cures that could be interesting and could affect early clinical and preclinical development. So it's really glad you brought that up. But, yeah.
I'd make one comment, and I'm wondering, Mike, Catherine, if you'd invited Peter Marks, one of the FDA members, to come to this meeting, because I think they'd be very interested in coming and listening and learning. Maybe a European regulator, they'd love a trip to California, frankly. So it's a good way of starting that interaction. And so I'd really, and if you can't get through to them, I know some of us on the panel have connections, so we could probably facilitate that for you as well.
We'll write that in the minutes. We've reached the end of our time. You want to wrap up the Go ahead.
What I've seen with companies taking these novel genetic therapies forward is that it's really hard to take such a valuable investment and put all your chips on one regulatory, right? So there's geographic, the way you manage that risk is, I see people diversifying. Let's go to America, Canada, Brazil, UK, blah, 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 Australia. If you had to give me 2 countries each, other than the US, to prioritize, not representing your companies, just as personal individuals who are experts in the space, what are the 2 geographies in addition to the US you think we should? Because we are an army, but we're a retired army. So I can't do every regulatory agency on Earth. So how can we prioritize? Just 2 each.
Sure. I mean, I'll start. I would do UK and Germany.
Yeah.
And the comment I'd make is, the company should build the interaction with these countries in parallel with doing the pre-IND meetings with the FDA. Because the package it's not quite the same, it's virtually the same. And I usually like to have one of the other countries' discussions first, almost as a practice run for the FDA, who tend to be tougher. So, UK, Germany, but there's several other countries. I could mention more that are pretty good as well.
I would like to say UK and Japan, but I also want to quickly make a plug because it is so underrepresented. I know, it's a conversation I was having with Martha. There is, and Vicky, where's Vicky, about the inequity of the Global South. And so just honestly taking a look to seeing if there is any feasible way to conduct at least one of your study sites in Latin America or another country where you have not gone, if you know the regulatory environment. And we know that there are countries right now that are doing a really good job. I see Carlos nodding in Argentina. So I would just add that in. That's all, off the record.
I think just 2 things I would want to add before I answer. I think it is worth thinking, I guess the priority and which agency you're going to first when you think about that. So some agencies, once you have engaged with FDA, they almost will just defer to what FDA says. So I think that affects which ones you go to. And then, as you're thinking about clinical trial preparedness and clinical trial startup, you also want to not just be looking at the regulatory environment, but actually the site startup environment and how long not only does that regulatory interaction take, but then how long do the hospital negotiations within individual countries take, because that can be a very, very slow process. In some countries, you can do some of that in parallel. So other countries, in addition to what Parissa and Suyash said, that I would consider would be Australia, UK again, and Canada too.
This side.
All right, so I'm not going to name names in terms of companies or countries, but I would say the consideration isn't just the regulatory agencies, also how can you mobilize patients and families in any jurisdiction. I think getting clinical supply to the country is actually more difficult than it sounds. So it's something, and it varies country to country. So I would just classify it as with some of these more bespoke genetic modalities, going somewhere other than the FDA is actually probably the expectation.
We're running out of countries. I think I would,
I didn't name a country.
we think of the EMA as well. So the European countries, of course, Japan, because you've got to test it on Japanese patients for approval there. And people have had success in other diseases of moving very quickly in Australia. So that's what I would look at.
I agree. We're running out of countries, but I would say echo that the UK, I've been in companies where we've done that and it worked quite well. Australia. But I also think I know friends of our CEOs who are going to Georgia and other places like that. I think it does depend on where the patients are and where the facilities are, where the hospitals are and the sites, because it does make a big difference on how easily you can get the patients.
I think that was a great question to finish in front of this banner that shows the global need for treatments and then the desire to bring it global.